this one. I believe you should see it now. Okay. So, what we mark here is that we again see a sample of mammary gland. Infiltrating ductal. Бабината ти дуктален. Това е нищо. Това е микст вариант. Това не е. Не. Това е с смесено. Говорена си в миналата. Чакай. So, what we observe here is parenchyma of the mammary gland, which is grossly appearing. They have margins of mature fat tissue here and there, and where in thick stroma, which becomes a little bit more loose around clusters of cells forming ductal structures here and there with papillary growth here. And here they're more compact. Medonecrosis, because they are positioned right at the center of the gland and they appear to be made by the squamated cells. Later, what we observe here, or why do we call it infiltrating? Because we see multiple of these nests of cells which are looking compact and attached to each other, and they will lose this attachment if it's a lobular type of carcinoma, but this is a ductal one. So this attachment appears here again. Although they look like compact clusters of cells, they pretty much stay together. Some forms of sheets might be observed at the periphery of the carcinoma, but to be sure whether it's lobular or ductal carcinoma, you should suggest to the professor that you will make immunohistochemistry staining for adhesion of the cells. And that, by my suggestion, would be the immunohistochemistry staining known, I will write it down here, cadherin. So that you will differentiate whether it's ductal type of carcinoma or lobular type of carcinoma, or whether it's infiltrating in sheets like Indian file pattern, because cells a little bit look separated at the periphery of the slide, or whether they keep the adhesions with each other, and it's more like ductal infiltrating. Here we also have necrotic structures, again, in vessels, and take a look at the stroma which appears around the glands. This is more loose stroma, which is rich. Did it disappear? Yeah, it made a mistake also here, so I sense it disappeared. Yeah. So, yeah, we recognize it by the look of the cells. Take a look at these. We should see two populations of cells in a ductal type of carcinoma. I'm searching for appropriate field. Usually, in normal acinar structure in the breast, from the lobules, we should expect two layers of cells which are positioned over a basement membrane. Those are the myoepithelial cells and the epithelial cells, which are positioned more apical. So here, we're supposed to see, again, two types of cells, some with more dispersed chromatin and darker cytoplasm, and others which appear foamy with more light cytoplasm and centrally positioned nucleus. So we see two populations of cells which are making this type of carcinoma. As far as you can see, it's not monomorphic. I'll search for more appropriate population to show it to you. There you go. Now you see a cluster of cells which are having two specific types of morphology. That would be the ones with foamy cytoplasm and centrally positioned nucleus, and some with more dispersed chromatin and more pinkish cytoplasm. These are actually generated from the myoepithelial and the epithelial cells. So that's how we recognize it. We won't see any normal structures here. Let me check out here. Maybe these appear a little bit more dilated, but the cells are having nuclear ATP. So I would not prefer to give information for this one. These are vessels. And yes, this is already a tumor. Most of it here of the slide is tumor, and others are with severe dysplasia. Here, there's some, no, these are not benign. Again, starting to be either with apocrine metaplasia or with already change into tumor cells. So I don't like these also, but a little bit more demonstrative about the two layers of cells. Take a look at the foamy, the epithelial, the myoepithelial, and the basement membrane. That's where it starts. And later, most of the slide is tumor, which is not well demarcated. It's infiltrating by nests of cells. I myself, looking at this type of tumor, I'm completely not sure whether this one is lobular or ductal. So I do not know who wrote this one to be infiltrative, ductal breast carcinoma, because they did not perform, or perhaps they performed immunohistochemistry, and this showed them there is there is actually preserved adhesion in between the cells, and they're staying in packages together. But if it's by this picture only in hematoxylin eosin, my suggestion would be mixed type, lobular ductal. Take a look at these sheets. They're appearing to be cell after cell after cell going next to each other and infiltrating in the surrounding tissues. Okay, I agree that's infiltration, but the manner of infiltration is also important. It should be in nests or in clusters of cells. It should not be this separated. So I would definitely perform this immunohistochemistry to make sure that it's ductal carcinoma. I suggest you tell it to the professor on your exam. His questions might be tricky. He already examined you. You know how he performs. So be really careful about your diagnosis. I myself am careful with him. Anyway, so next thing would be liver changes in chronic myelogenous leukemia. Let me share it with you.